Hello. What we're going to do today is we are going to finish off B4 and we are going to start C4. And I will do a separate video for the genetic problems. Last time we went through most of these non-Mendelian genetics, or exceptions to Mendelian genetics. Linked genes, so you need to know what they are. You need to know how they violate the Mendelian genetics, and make sure you know the examples. Today we're going to finish off the blood type and look at polygenic inheritance. X-linked, we're actually going to do that in C4. So we may not get to that today, but we did do it in the lab, in the human genetics lab. So let's continue on with our blood. Multiple alleles, A and B, but now we're going to go through the positive and the negative. The Rh positive and negative factor are inherited totally separately from the A and the B and the O blood types. So human Rh factor, positive or negative, again, it's a completely different system, not related. Phenotypes, we have positive and negative, positive, negative. The negative is recessive. So the negative can be masked by the positive. So in the heterozygote right over here, you can either write it like this, R and H's, or just R's. So in the heterozygote over here, that negative is masked. So that means you can have two parents that are positive, and both of them, if they're heterozygous, can have that Rh negative gene, and those two parents, then both being positive but heterozygous, could have a child that is Rh negative. So in this case, you either have it or you don't. Notice here, here's the antigen. No antigen. So these are positives, negatives. So those over here, look over here, with the positive, no antibodies. Negative, look, they have antibodies. So look at B. Neither Rh positive nor Rh negative individuals normally actually make those antibodies to the Rh factor. Rh negative individuals are going to make them when they are exposed. So normally these are not going to be here. That's different from the A and the B blood. In the A and B blood, the antibodies are going to be automatically there. But with this, these antibodies right here, again, they're not normally going to be there unless they're exposed to the Rh positive red blood cell. And that's going to be really important during pregnancy. And it's because of this that in the past, the requirement to getting married was that you had to get a blood test. And that was to see if it had positive and negative. We'll see what will happen if one's positive and one's negative. So let's say the father is Rh positive. And the mother is Rh negative. And her first child is Rh positive. Lots of times with the first child, everything will be fine. But remember, the mom is not yet going to have anti-positive antibodies unless she's been exposed. But when the baby's developing or being born, especially during delivery, there can be a mixing of the blood. So now the mother will be exposed to the positive blood. So her negative blood exposed to the positive blood. And then her body will begin to produce anti-Rh antibodies. And then if she becomes pregnant again, and her child is Rh positive, then the anti-Rh antibodies can cross. So let's look at here, positive, negative, that was positive, she's been exposed, she produces anti-Rh antibodies, those are those green things, becomes pregnant with another one. Now those antibodies can cross the placenta and cause damage to the baby who has positive blood. And the result of that is something called HDN, hemolytic disease of the newborn. Due to hemolysis, lysis we know that means is split, right? Hemo and blood, right? Blood, heme. Remember that from my hemoglobin when we talked about different types of proteins? So this is going to continue after the child is born. So the antibodies that the mother produces attacks the antigens and the red blood cells in the fetal circulation. They break down and destroy those red blood cells. And this has caused a lot of trouble. It can range from mild to very severe and fetal death even. Anemia, jaundice, even heart failure, brain damage. When the disease is mild or severe, there are many erythroblasts in the fetal blood, so that's why they can also call it erythroblastosis fetalis. Or they can also call it Rh disease. That's estimated to occur in about 2 out of every 100,000 births in the U.S. The baby can be born with severe jaundice, characterized by a buildup of something called bilirubin. This buildup of the bilirubin causes characteristic yellowing of the skin, whites of the eyes, so it causes jaundice, right? Orangish, yellowish. Bilirubin is a product of the breakdown of those red blood cells. Normally the liver processes the bilirubin and sends it to the intestines. But in the baby with HDN, the liver may not be able to keep up with all the red blood cells being destroyed. So the result can be a serious buildup of bilirubin and severe jaundice. And light therapy is usually used to treat it. You just put them in the sun. And they actually use blue light. They use the blue light because that breaks down the bilirubin the best. These are called blue babies. I remember when I was younger, my younger brother had this. I was so excited because they told me that my brother was a blue baby. I thought he was going to come home and be blue. I thought it was pretty exciting. But instead, just this. Look at this picture over here. They just put him under the blue light. When he got home, they had to keep him in the sun. And it's actually pretty easy to prevent this from happening. And again, that's why they had the blood test. If a blood type incompatibility is found, like positive and negative, preventive measures can be taken to avoid this, the HDN. The mother can be given injections of proteins that will prevent her immune system from producing those antibodies, so they'll no longer attack her future baby's red blood cells, or even if she's pregnant at the time. Or even in her first child, if they have this condition, she can be given the shots so that she'll not produce the antibodies against her next child. We will do some genetic problems with this in the future. Having gone over the multiple alleles for the blood, notice here, actually blood types are a lot more complex than we thought. Over 20 blood group systems and 7 antigen collections have been defined. So look at this. It's a lot more than we thought. Here's just the ABO here. Here's our Rh positive and negative. Others they found through giving transfusions what they thought were supposed to work, but they didn't. Look at this. All these different systems. So blood type is actually very, very complex. Alright, let's go on to our next Mendelian exception. Polygenic inheritance. Now this is like the opposite of pleiotropy. Pleiotropy, one gene had many effects. Poly means many, many genes. Many genes have one effect, so they're opposite. So polygenic inheritance, multiple independent pairs of genes have similar and additive effects on the same trait. Numerous genotypes are going to occur, and the more genes involved, the more genotypes and the more phenotypes we're going to have. Number 4, the offspring exhibited continuous variation in phenotypes. Example, as many as 60 genes account for skin pigment in humans, seed color in wheat, human height, eye color, tail length in the anolis lizard. So these are controlled by many genes, and they have that one effect: human height, human eye color, tail length. So let's actually go ahead and look at an example. So let's look over here. It says this is for 3 genes for skin color, but we know there's a lot more. But look, all these different possibilities of different genotypes and different phenotypes. We know at least 3 genes control eye color, but we know there are more than that. And we're going to look more at eye color. So there's going to be a great range of traits, continuous range. Again, many polygenes for one effect. A lot of this is just for your interest because many people, many students, always ask me about eye color. So you need to know that eye color is polygenic. And before we can actually understand eye color, we need to see what it looks like, what the eye looks like. So the eye color is determined by the amount of pigment called melanin in the iris. So this is what we're going to be focusing on. The iris. So the amount of pigment is determined by, we know, several genes that control the pigment production. Eye color genes, the enzymes they produce, are going to direct the amount and the placement of the melanin in the iris. For those who are born with blue eyes, a lot of Caucasian babies are born with blue eyes. To the time of birth, they don't have any melanin, so no melanin produced. So the eye color actually may change. After birth, maybe the melanin will begin to be produced. Babies of other ethnic origins like African, Asian, Hispanic, Native American, because their eyes are born with pigment already present in their iris, their eyes tend to be brown or even black. Pattern in the pigment, the pattern which the pigment is deposited is also determined by genetics. Some people have these like beautiful eyes. They have all these rings and clouds and stripes. And we don't really understand how that works. So we mentioned blue eyes. There's no pigment in the front layer. Now we need to know also that there's 2 layers in the iris. And that is going to be really interesting and really helpful to understand that there's 2 layers. Because there's 2 layers, we get a lot more different types of colors than if there's just 1 layer. So there's a front layer and there's a back layer. And blue eyes, there's no pigment in the front layer. There's only going to be melanin in the back layer. I have blue eyes, but my eyes are not really blue. Just like the sky is not really blue. So just like the sky is not really blue, blue eyes are not really blue. The reason why the sky is blue sometimes is because blue light, those wavelengths, are going to be scattered by gas molecules or atoms. So they're just the right size to scatter those blue wavelengths. And if you have white, like sometimes it's like hazy, there's a lot of particles, that means all the different wavelengths are going to be scattered. So it looks blue because those blue wavelengths are scattered in your eyeballs and it looks blue. Again, because of the wavelengths coming in, no pigment in the front, we have selective scattering of those blue wavelengths, and we have blue eyes. So again, appears blue due to selective scattering of the light, in this case the transparent iris. The green eyes, dark pigment distributed. Alright, so we have dark pigment is in the front layer of the iris. Of the background, it appears kind of gray. Wrong one, sorry about that. Green eyes. Small amount of melanin is deposited in the front layer and in the back. Alright, now to the gray eyes. Gray eyes, the dark pigment is distributed in the front layer of the iris, so it's going to appear gray. Okay, brown eyes. Brown eyes have so much pigment in the front that it completely covers up the blue behind it. Purple or violet eyes, well, we're not really sure whether there really are such things. Hazel eyes, blue-green, gray-blue, all these different variations, all these different patterns. We're really not sure how this happens. Albinos, no pigment at all in either of the layers. The iris appears pink or red, and the reason why is because of the reflection of the blood vessels in the back of the eye. So you can see the blood because there's no pigment. Alright, here you need to know this. So you need to know that eye color is polygenic. And you need to know the 3 genes involved that we know. We know that there are more. You need to know that brown is dominant over green and blue. And green is dominant over blue. So brown is dominant over green and blue. Green is dominant over blue. Alright, so let's assume that we have complete dominance. And let's just talk about brown eyes and blue eyes. Let's say big B is brown. So big B is the dominant allele, brown is the phenotype. We have 2 genotypes possible. Homologous dominant, heterozygous. So we're going to say little b is going to be blue. So complete dominance, this will be homozygous recessive. So let's say we have 2 parents, and they are both homozygous recessive. You'd expect all the offspring to be homozygous recessive. So if both parents have blue eyes, we would expect in complete dominance that every single offspring, and if they had 1,000 children, they would all have blue eyes. That's what we would expect. So the question is, I should cover up the answer. Hold on a second. Can 2 blue-eyed parents have a brown or green-eyed child? Let's go take a look. So we have 2 blue-eyed parents. Could they have a brown or green-eyed child? Or we're just talking about brown. Based on this, we'd say no. And this is what we were always taught in school. So if we're taught this in school, and we have husband and wife, they both have blue eyes, and then produce a child that has brown eyes. Alright, and the man's gonna say, honey, what happened? How come our child has brown eyes? And she said, I didn't do anything. And actually, for reasons unknown, they can. I mean, there's a lot about eye color that we don't know. So there's post reports in the medical literature which 2 blue-eyed parents had brown-eyed child. This is more than once. Currently, there's no explanation, but genetically, they were the parents, and genetically, that that was their child. So there must be some kind of unidentified eye color genes involved that we don't know about. It goes in line with this. Can paternity be determined by the eye color of the child? We now know it can't be. We once thought it could be. But we know that eye color is not the simple Mendelian dominant recessive trait we once thought it was, just like I showed you in a genetic problem we just did. It's multifactorial, polygenic. We don't know all the genes that contribute or how they combine to give different colors. Could you have mutations in genes? Maybe the thing called things are turned on, turned off. These kind of interfere with production, transport, deposition of melanin, produce more melanin, reduce the amount of melanin, and you can have all different kinds of eye color. So this can result in eye colors that are unexpected given the parent's color, as far as we know at this time. Blue-eyed parents can have a brown-eyed child. It doesn't usually happen, but it is possible. Next question, why do some people's eyes change colors? That could be due to increased or decreased pigmentation. How can a person have two differently colored eyes? For example, one brown eye and one green eye, or one brown and one blue eye. This actually is called heterochromia iridium. So the fact that you're having two different colored eyes, it's called heterochromia iridium. And this is caused by differential deposition of the melanin into the iris of one eye versus the other, so one has more melanin than the other one. And it could be pathological. Could be really no problems. Here are some examples. Blue and brown, hazel, all different kinds. When we were growing up, we had a Siberian Husky, two different color eyes just like this. Her name was Sonia, and somebody stole her. But she had two different colored eyes. How can someone have one green eye and one eye which is half brown and half green? No one seems to have the answer for it as yet, but in the cases that they have figured out, it was due to some kind of injury in one of the eyes. And so because of the injury, the part of one eye was either producing too much pigment or not enough. Next we're going to be looking at something that's going to lead us right into C4, our next handout. Something called phenotype plasticity. You don't need to know these words, penetrance and expressivity. Just know that your genotype only specifies your potential. So, you have your DNA, how it's expressed, depends on the genotype, but also the external environment, which is what we're going to be talking about. So let's look at the picture below. So all these beagles, they have the exact same DNA. Look, they all have different kind of spotting. So the DNA, the genotype was just their potential. So something else obviously is going to be involved. So let's look at this example. So we have these hairy guys, and they all have the exact same genotype. And so we're going to be expecting them to all look exactly the same. So that's what we're expecting. 100% looking exactly the same. They have the same environment and the same genotype. Some cases, look right here. Some have it. Some have the hair, green hair, and some don't have any. They're bald. You're expecting this to have exact same environment and genotype. And this is just showing you that there's a range. There's some that are really hairy, medium hairy, and not very hairy. let's go on to our next handout, C4. In C4, we're going to talk about something called epigenetics. This is a big growing field, and this is going to show us just what we ended up in the last handout. That your genotype is only your potential. It's only your potential. Let's look at the word epigenetics. Epi, right here, epi. That means above, on top of above, as well as beyond. So, epigenetics. Let me put beyond here. Epi, beyond genetics. So there's something beyond our genetics. So something beyond our genetics. Right? So looking at epigenetics is study of heritable changes in our phenotypes. So we can actually inherit changes in our phenotypes without changes in genotypes. And if you remember, well actually we haven't talked about it yet. I'll get to it later. So there's some type of external modifications to the DNA that can turn genes on and off. The modifications are not actually going to change the DNA sequence. They're just going to change how the DNA is read. So they're just going to change how it's expressed. So actually we're learning more and more things that can affect our DNA and the expression of our DNA. So that field is called epigenetics. Some things that can affect the expression of our DNA are what we call epigenetic factors. Histone modification, DNA methylation, and microRNA. DNA methylation, addition of a methyl group or a chemical cap to part of the DNA molecule. So basically that is just going to prevent genes from being expressed. So the key thing is that if there's something that's preventing genes from being expressed. It's not changing the DNA, it's just not letting it be expressed. Histone modification, we'll talk about that, and I'll show you a picture to understand this. The proteins that the DNA wraps around, without these, DNA would be too long to fit inside cells. So in order for transcription, remember transcription, the messenger RNA comes in and has to read the DNA. But the DNA is wound tightly around these like little spools. So the histones have to relax. They have to let some of that DNA out so that it can be read by whatever is going to read it, like the messenger RNA. So there's going to be modifications that are going to relax these histones to make that DNA accessible so that transcription can take place and then translation can take place and the proteins can be made. Usually when we think of our DNA, we think of this ladder thing here. See it in the form of a ladder that's twisted like a double helix. But it's not really like that. Right? It has to be wound around these histones. These are histones. They get wound and wound around them. They're compacting the DNA. You can get miles and miles of DNA in each nucleus. The DNA is really long. So the only way we can actually have so much DNA is to wrap it around these histones very tightly. So DNA is wrapped around the histone proteins. They're going to form nucleosomes. Histones are going to be important in the regulation of gene expression. They're going to be involved in turning genes on and off. So nucleosomes are going to be a section of DNA wrapped around a cord of 8 histones. And they're going to prevent DNA from being tangled up. So those are the important things for us right now. So DNA methylation, you're just going to put a cap on it, prevent certain genes from being expressed. Histone modification, here's the histone OCT8. So again, histones are those proteins that DNA wraps around it. So we need to get it to relax to make the DNA accessible so that it can be read by the messenger RNA in transcription. Here's an example over here where it's wound and it's not being accessible. The closed chromatin, this causes longevity. Open. See how it's getting relaxed? It's tight, it's relaxed. That causes aging. So the DNA is the same, but what's different is how the DNA is being able to be read. This one is being read, this one cannot be because it's wound up around the histones. MicroRNA that can bind the RNA, can break the RNA down so the RNA cannot be read to make the proteins. There are histone modifications, DNA methylations, so that can, all these three things can change how the DNA is read. It doesn't change the genes, it just changes how they're being read. So some of them are going to interfere with transcription. If they interfere with transcription, they're going to change which genes can be expressed. So they're changing gene expression, not the actual DNA. So DNA sequences are not changed, just the way the DNA is transcribed is changed. Remember we went through that in test 2. DNA can never ever leave the nucleus. So messenger RNA gets the message from the DNA, takes that message out to the cytoplasm, and uses that sequence, uses that message to make the proteins. So DNA sequences are not changed, just the transcription is changed. Because the transcription is changed, this is going to change the way the genes are going to be expressed. And these changes are heritable. And you can think of the genome as hardware, and the epigenome as the software. So the epigenome is more flexible, and it's going to be changed. It's going to change what gets passed on from parent to offspring, and that can be influenced by the environment. Okay, let's look at some examples. Here's an example of an experiment to figure out if these changes could be heritable. So they had these mice, and they would give them the fragrance of cherry blossoms. And every time they smelled cherry blossoms, they would be given terrible shocks. So very quickly, they learned every time they smelled cherry blossoms, they were going to get shocked. So whenever they even have the faint glimmer of a cherry blossom fragrance, they begin to tremble and shake and act really nervous. And their offspring exhibited the very same behavior. They had never got shocked or anything like that. But whenever they would smell cherry blossom, they would begin to shake. Just like the parents that had the shock treatment. Another example, mice running a maze. In this case, instead of being shocked, they actually were rewarded. So one study involving the use of mice showed that the first generation had to run a maze. It took them several months and actually learn how to go through the maze to get the prize. So it took them a long time to actually learn it. And then their offspring, right, so their offspring could actually run the maze within seconds of first being introduced to it. So that's pretty amazing. And what about these young children? They just know how to use a smartphone. They just know how to use computers and iPads. No one even teaches them. They just know how to move their hands and just swirl the smartphone to get to the next screen. It's amazing. You just know how to do it. So there's a lot of different things that can cause methylation, etc. Diet, lifestyle choices, behaviors, stress, development, drugs, emotions, thoughts. And we're learning more and more on what these things can do. The genetic traits are a product of your DNA and your environment. Environmental conditions and life experience of parents, grandparents, even great-grandparents, maybe even great-great-grandparents can really flip on and off switches in the genes in their egg and sperm, or the genes of developing fetuses in pregnant women, and they can actually change how it's expressed. Evidence suggests smoking, overeating affects your DNA, or how it can be expressed. It causes those affecting obesity to become switched on. Those carrying messages for longevity become switched off. That means, in addition to the self-harm that can come from eating too much or smoking, these lifestyle choices may predispose a person's offspring, and even future descendants, to disease and premature death. Now, this doesn't pass on bad things. It also passes on good things. So if you're eating well, you're healthy, you're doing exercise, etc. That also gets passed on. So not just bad things, but you can cause your offspring to inherit healthy things. That's good news. Many of the studies in humans and epigenetics was done through identical twins, because identical twins have the same genotype. So here are some pictures of myself and my twin sister. That one's me. That's Rebecca. That one's me up front. That's Rebecca. There. Look at the recent pictures. Right here you can see I'm close to the camera. So why are identical twins different from each other? Identical twins are not exactly identical. The correct term for identical twinning is monozygotic. So let's talk a little bit about identical twins, because I think it's very interesting. I'm sure you do too. The zygote splits into two parts after conception, resulting in the development of two individual embryos. Because the two embryos are the result of a single egg-sperm combination, they have the same genetic origins. They have the same DNA, and they are the same age. Identical twins are the same age, based on conception. They have the same name, but they don't look the same. They don't act the same necessarily. In the case of identical twins, what triggers a single fertilized egg to result in two embryos? What is the probability of having identical twins? They don't really know. Right? They don't know. But, contrary to public opinion, identical twins do not run in families. Therefore, twinning is not related to genetics. And it's pretty much the same over all ethnic groups. During the first 4 days after fertilization, a huge fertilized egg, zygote splits into many smaller cells suitable for building the eventual baby. Sometime after the 4th day, about 30 of these similar cells are hanging around. The spaces between them are filled with pools of clear fluid and gather together to form a hollow cellular ball called a blastocyst. An inner cell mass forms inside. This is the stuff that an embryo will develop from. Now look at day 1. Zygote is split into two smaller building block cells. If the two cells, we're going to be looking at different kinds of identical twins. If the two mass splits into two clumps in day 1, okay, day 1. So if the twins form in day 1, these twins are the most identical. These always have separate placentas. because they split so early. And what's interesting is, because they have separate placentas, doctors often mistakenly these most identical twins as fraternal twins, even though they are the most identical twins. Most twins, identical twins, form between day 4 and 5. And these could be mirror images. So mirror image twins. Sort of what my sister and I are. Day 9 and 10. They're going to share the same inner sac, which is not necessarily a good thing because the umbilical cords can twist together. Day 13 to 15. Cells forming the two embryos don't totally separate. And this we get twins born joined together. About 3 in every 1,000 deliveries are identical twins. Dizygotic twins are fraternal twins. They're just 2 eggs fertilized by 2 separate sperm. 1/3 of all twins are identical. So fraternal twins, dizygotic twins, are twice as common as monozygotic. So day 1 to 3, diamniotic, their own placentas. Day 4 to 18. So we have different kinds of twins depending on when they separate. After day 13, conjoined twins. Here we are when we're a little bit younger. Why do identical twins have physical differences if they have the same DNA? And that is because of epigenetics. So identical twins look very much alike because we have our own DNA. But because of the environment, that can cause epigenetic factors to turn things on, turn things off, environment, etc. All these things can have a big impact on how our DNA is expressed. So different things that can happen during your life, like maybe one of them got really sick, or one of them had a different job, one did a job where they did a lot of physical activity, one of them did a desk job. That's going to affect their phenotypes. So this person wrote, think of it this way, when I bake an apple pie, it turns out a little different each time. Shapes of the apple pieces are never quite the same. Stove may be a little hotter. Add slightly more sugar. Distractions. Phone ringing. Exactly when I'm trying to take out the oven the pie out of the oven. So use the same set of instructions every time. Each pie is still unique because differences in the environment make it impossible for me to follow the instructions in exactly the same way. Identical twins are kind of like 2 pies made from the same recipe. They will always have some physical differences due to differences in their environment. And it could be actually in their environment when they're inside the womb. So they're developing, maybe one will get taller, way more. So again, they're doing a lot of research with identical twins to see if something is controlled by DNA, one is controlled by the environment, one is interactions. Because again, identical twins do have the same DNA, for the most part. So we know since identical twins are different, maybe even different in height, something else is going to be involved. Here we are when we're babies. When we're babies, we look pretty much exactly the same. and they had little bands that they put around, my mother put around our wrists. And one time, she gave us both a bath at the same time, and she took our little wristbands off. And then she got a phone call, and then she got distracted of who was who, and she guessed. So I could be Rebecca, and Rebecca could be me. There's an experiment that was done on two NASA astronauts, Mark Kelly and Scott Kelly. They look pretty much the same. So they wanted to see what would happen if Scott spent an extended period of time in space. So they did all these tests and things, tested all these different kind of things. And after I got back from spending a long time in space, they did some more tests. They found some differences in the mitochondria, I'm not sure what they were, but other different differences in the genes. They actually saw that about 7% difference in the gene expression before and after. So the extended space environment changed Scott's phenotype. Perhaps because there was less oxygen, more carbon dioxide in that atmosphere. This also from space travel. So it affected how the genotype was expressed. So they had the same genotype still, but the genomes were identical, but the expressions were different. So here's Scott on the left, Mark on the right. Okay, let's continue on with C4. Another thing that is really important in epigenetics are your thoughts, your choices. When you're choosing, you're actually changing the physical nature of your brain. Every thought you're thinking affects every cell of your body at quantum speeds. That's faster than the speed of light. And your thoughts are going to be creating chemicals. Chemicals can turn things off, turn things on. So your thoughts are quantum signals inside your brain. And your thoughts can go into the egg and sperm and stay there for the next 4 generations. So they're doing experiments with this, that they're learning how to control their genetic expression through your thinking. And lots of times when your previous generations have passed things on to you through epigenetics, they actually have to be activated by quantum signals from your mind, like genetic predispositions, depression. And you think, my mom's depressed, I'm going to be depressed. You generate a signal that activates that. So when you are born, those things that are active, they become dormant. And you can turn them on or turn them off. So you can unzip or destroy those things from past generations. And they call that reprogramming. So it may be possible to pass down epigenetic changes. Again, talking about good things. You can change things by passing down good things. So this can be a pretty exciting time. Let's give an example. Alright, let's say we have some fraternal twins born and they're sisters. Alright, let's say one of them is really pretty. That's the pretty one. Not all that pretty to me. That's the pretty one. That makes me ugly. Pretty. There's the pretty one. And here's the ugly one. Okay, they're sisters. Here's the pretty one. B is the ugly one. Okay, they are fraternal sisters. And they have a predisposition towards depression. So it's not turned on yet. Alright, they can turn it on or turn it off. And let's say they're born into a family where looks are really important. This one, the pretty one, she's going to get a lot of affirmation. Oh, you're so beautiful. And let's say she's really talented. Oh, you're so good at this, and you're so good at that. And her poor sister, not talented, rather clumsy. Doesn't look good. This one right here, they both of them have the same genetic predisposition towards depression. This one is less likely to turn it on than this one. So they both have it in their epigenome to be depressed. So they both have it. So this one is going to be more likely to activate that one. So that one will actually be activated toward depression. And this one will not turn it on. I mean, they could. I'm just saying, setting up this scenario. Maybe she's mocked. This one over here is mocked. She's yelled at. She's clumsy. Spills things. So this one's probably going to activate their depression, but this one would be less likely to. And it could be that neither of them do, or both of them do. So even though they have it, they are born with it inactivated. It is dormant. They turn it on if they want, or keep it turned off. So it's not just your genes, also your environment. So here's some other environmental effects on your phenotype. Things that can influence your gene expression. Water. How much water you have. So water, temperature, light, nutrition, chemicals, infectious agents, other organisms in the environment, etc. All those things can affect your gene expression. Here's an example of fur color in Himalayan rabbits. This one right here is the normal pattern. Black ears, black nose, black feet, black tail. And if the temperature, and that's 25 degrees Celsius. If the temperature is more than 30, it's going to be all white. And if they have it at the normal temperature, but they put a little ice pack right there, that's going to get dark. Human height, we talked about that. That is polygenic. So your genes are going to set your certain limits. Those are going to be your potential for your phenotype. So if someone has the DNA to be tall, but they don't have nutrients, they don't have very much nutrition when they're growing up, they're not going to reach that potential. So what phenotype an individual develops within these limits depends on the environment. And the human height has increased about 1 inch per generation over the past 100 years. It's probably not because of a change in the genotype, but maybe because of the environment, better nutrition, better health care, more time to exercise. Here's some other examples of a phenotype is controlled by the environment and the genes. Skin color. You can go out in the sun and get tan. Don't go in the sun and get white or a lighter color. Coat color, influenced by the temperature. It's cold, snowy, turn white. Summertime. Hotter, change color. Over here you have hydrangeas, influenced by the soil pH. So over here, in acidic soils, they're going to be blue, so that's acidic. Alkaline, basic, they're going to be pink. Octide grow hundreds of years and they always die. Okay, now change the subject. We're going to do one more of exceptions to Mendel, and I mentioned that we're going to go through this earlier today. Alright, so sexual inheritance. Genotypic sex determination. So talking about genotypes. Y chromosome mechanism seen in humans, where it is the Y chromosome that determines the gender of the offspring. So any individual that has the Y is genetically a male. If you don't have it, so you lack it, genetically a female. X chromosome. Main factor in sex determination will be the ratio between the number of X chromosomes and the number of sets of autosomes, so we don't do that. In this case, and these are different types of genetic sex determination. In this case, this type, Y chromosome has no effect, but it is required for male fertility. Here, Y chromosome, Y determines it. In this case, it's the ratio between the X chromosome and the sets of autosomes. Y chromosome is no effect in gender determination, but you still need it. Otherwise, the male will not be fertile. Some lizards, many snakes, and in birds, it is the female which is heterogametic, has differing sex chromosomes. So let's go back up to the Y. Up here, Y chromosome is the male that determines the gender of the offspring. The male, XY, we call that heterogametic, so they're not homologous. Here, let's talk about birds. It is the female which would determine the gender of the offspring, because the female would be the heterogametic. And sometimes you write it ZWs, or male would be ZZ. That'd be comparable to XY and XX. Let's review autosomes and sex chromosomes from our lab. Humans, 23 pairs of chromosomes, 22 pairs are autosomes, 1 pair of sex chromosomes, XX and female, genetic female, XY, genetic male. So XY system, right here, male XY. When sperm cells contain a Y chromosome, females are XX. XO, certain insects. Male occurs when sperm cells contain no sex chromosome, female XX, when sperm contains an X chromosome. ZW, that's the one I just talked about. Opposite sort of the XY, that the females are heterogametic and the males are homologous. Then we have something that's totally different. Haploid diploid systems, birds and ants, they go back and forth. The males are the haploids. You may have learned all about this in grade school. No sex chromosomes, male develops from an unfertilized egg. I saw videos about this. The female is the diploid. So males are all haploid. And they just develop from unfertilized eggs. So since there's no X and Y and no two sex chromosomes, they are haploid. Females are diploid. Right, so they have a male and a female. And they're going to develop from fertilized egg. The Y chromosomes, pretty much what they do is they make the male a male. They don't really carry much information. Whatever's on the X chromosome, talking about the X-linked chromosomes, that's going to show up on the male. We are going to go through genetic problems like this. We went through this in lab, so I'm not going to go over this a lot right now. but when we do the genetic problems, I will come back to this. So in Y-linked, the males determine the gender of their offspring. So some families have more boys than girls, or more girls than boys. It's not genetic, it's a physiological response. Maybe the Ys are faster swimmers or something. And remember these two traits from our lab. X-linked recessive, red-green color blindness on the X chromosome, hemophilia on the X chromosome, but they are not related to gender. So again, we will go through examples of these when we go through our genetic problems. Now we're going to go through some phenotypic sex determination systems. Where the X's and Y sex chromosomes are not going to be involved, but it's going to be the environment that determines the gender of the offspring. Let's look at this example, the marine worm Amelia. Free-swimming larval form, it is sexually undifferentiated. And where it settles down will determine whether it becomes a female or a male. So if an individual settles down alone, it becomes a female. If it attaches to the body of an adult female, so there it is, attaches there, then it's going to become a male. In some lizards, turtles, crocodilians, the genes that control gender are dependent on temperature during a critical period of incubation. It's not having anything to do with the genotype, but the temperature at a critical time. So often only males develop if incubation temperature is optimal. Females, only females if it's very high or very low. So perhaps if it's in between, they'll get both. So again, these two examples were phenotypic gender determination. Nothing to do with their X or Y chromosome or any gender sex chromosome. So humans with the Y chromosome mechanism. In the first few weeks of fetus development, your baby's internal and external genitalia are going to be the same, regardless of whether you're ultimately going to have a boy or a girl. Which sex organs develop depends on the presence of the male hormone testosterone. So in humans, the default gender is female, and female will develop unless maleness is actively induced by the Y chromosome. At 12 weeks, most of this male differentiation has occurred, but they're still not completely formed. On the ultrasound, if your baby's cooperating, gender can be identified as early as the 16th to 18th week of your pregnancy. So it has to do with the testosterone that determines whether it becomes a male or a female. So our sex-linked traits, again one more time. X-linked recessive, sex-linked in males. You have the mutation in the X chromosome. These are the only two we're going to be looking at, red-green color blindness and hemophilia. In your lab book there are more examples, but we're going to just stick with these two. And I will be doing some genetic problems on a different video. We also have X-linked dominant. There's only a few dominant X-linked traits that we have identified. Faulty tooth enamel, dental discoloration. And they're going to follow the same sort of inheritance rule as X-linked recessive, except kind of opposite. Heterozygous females express the trait. You will not have to do any genetic problems with this. Y-linked inheritance, well, we haven't found any yet, but if there was one, we would be able to recognize it, because every single male would have it in this particular pedigree. Next, we're going to look at something called sex-influenced characteristics. So the two sex hormones, testosterone, produces what is considered maleness. Mustache, beard, Adam's apple, lower voice, muscles. Estrogen, breasts, higher voice. So you can be a genetic female and physically appear to be a male if testosterone kicks in your development inside the womb. Or you can be a genetic male, and if testosterone does not kick in, then the male genitalia, etc. will not form. Alright, let's get down to E. Cancer of the reproductive tract. What they do is they treat it with the opposite hormone. So prostate cancer, they'll treat it with estrogen. Uterine cancer, they'll treat that with testosterone. The hormones are going to be determining physiology and the phenotype. So sex influence characteristic. The relationship between genotype and phenotype is going to be different between the sexes, and our example is going to be pattern baldness in humans. Talking about this kind of baldness right there. It's going to be one pair of alleles of an autosomal gene that's going to be involved, and it is going to act as a dominant in males. So this allele acts as a dominant in males because of the higher testosterone, and it acts as a recessive in females. So males will only need one, but females will need two. And that's why pattern baldness is far more frequent among men than women, because women will need two. One from their mom and one from their dad. Sometimes in females, if they have this allele for pattern baldness, they may not know they even have it until maybe when they get to menopause, when the testosterone levels are a little bit higher, then the allele will act as a dominant because of the higher testosterone. But even then, they probably won't get bald like the male. Instead, they'll just have thinning of the hair. The frontal hairline will probably not recede. They're not going to lose as much. They're not going to get totally bald. Because of the higher testosterone, that's why females may get facial hair, whiskers growing on the face. So today we finished B4. We did at least half of C4. And next time, we're going to finish C4, do a little bit of beginning of D4, and do our genetic problems. So there's probably going to be only one more video. We'll see. So until next time, be well.